Welcome to this explainer. Today, we're unraveling something that is just absolutely magnificent. I mean, it's mesmerizing. We're looking at what is perhaps the most complex molecule in existence and almost certainly the most important, DNA. But I don't want us to just look at this as some dry biological concept, Instead, we're going to explore this incredibly complex molecule as a century-long detective story. The clues were literally hidden in plain sight, tucked away inside every living thing on Earth. But it took nearly 100 years of brilliant minds piecing together microscopic evidence to finally crack the case. So we're going to put this puzzle together, almost like an elegant microscopic papercraft model, to understand exactly how it makes you, well, you. Okay, let's dive right in.
To truly appreciate this mystery, we first need to grasp the sheer scale of the enigma inside us. We so often think of ourselves as a single, unified entity, right? But the truth is far more fascinating. You are actually a walking galaxy. Right now, as you're listening to this, you have about 50 trillion individual cells operating in your body. And almost every single one of those microscopic universes is actively guarding this exact same genetic instruction manual inside its nucleus. It's everywhere, all at once, constantly working to keep you alive.
And to really break our brains with the scale of this whole thing, just imagine pulling all the DNA out of every single one of those 50 trillion cells and laying it all end to end. This microscopic thread wouldn't just stretch across the room. It wouldn't even just stretch around the Earth. It would stretch from where you're sitting right now all the way to the sun and back again, an astounding 600 times. That impossible cosmic distance is currently coiled up inside your physical body right now. No way, right? But it's true.
And the sheer complexity perfectly matches that massive size. In almost every single one of your somatic cells, you have 46 chromosomes containing this tightly packed DNA. That structure is composed of 6 billion base pairs per cell. So to put that into perspective, if you printed out the 6 billion letters of this genetic code into physical books, you would literally need 10,000 books, each 1,000 pages long, just to hold the instructions for building you. The paradox here is just awe-inspiring. 10,000 thick books worth of biological code packed into a space so infinitesimal that the human eye can't even begin to perceive it. Now let's trace exactly how we figured all of this out, because this is where our detective story really kicks into gear. When people ask who discovered DNA, the names James Watson and Francis Crick usually get all the credit, right? But actually, scratch that, it's not entirely true. They didn't discover DNA itself. They just delivered the final act of a much older, much deeper mystery. The real story starts way earlier than the 1950s, and frankly, the truth is a whole lot messier.
Tracing this historical roadmap reveals that unlocking the secrets of DNA wasn't just one sudden eureka moment in a pristine laboratory. It was a multi-generational human relay race of science. It took nearly 100 years of painstaking research, absolute dead ends, stolen clues, and just brilliant minds across different countries to decode this puzzle. From 1869 all the way into the 1960s, piecing together this molecule was an absolute marathon.
The journey actually starts in 1869 with a Swiss biologist named Friedrich Miescher, and his method was fascinating, if, a little bit gross. He collected used, pus-covered bandages from a local hospital. He painstakingly washed the cells in warm alcohol and enzymes to isolate a gray, gooey substance he called nuclein. Miescher had literally just found the secret to all life on Earth, but he had absolutely no idea what it was. He just thought it was some weird gray goo. Today, we know it as nucleic acid, and we know it's built from 3 microscopic Lego-like pieces, a 5-carbon sugar called deoxyribose, a phosphate group, and one of 4 nitrogenous bases.
Fast forward a bit to the 1950s in London. Enter a brilliant young biophysicist named Rosalind Franklin. Her meticulous work provided the definitive magnifying glass that unlocked the whole mystery. Using a technique called X-ray diffraction, she fired X-rays at crystallized DNA to capture its shadow. Now, this wasn't just some simple tool. It was incredibly difficult, highly dangerous work that required peering into the invisible. Through this, she was likely the very first person to actually confirm the shape of this legendary molecule.
And here is where we find that classic stolen clue trope in our scientific detective story, which highlights a genuine historical tragedy. Franklin's meticulous, dangerous legwork proved the double helix shape and correctly placed the sugar-phosphate backbone on the outside of the structure. She even shared data that steered her competitors, Watson and Crick, away from a flawed triple helix theory. But the intense radiation exposure she endured likely caused her early death from ovarian cancer at just 37 years old. Because the Nobel Committee doesn't award prizes posthumously, she was disqualified from the 1962 Nobel Prize.
Her rivals, who built upon her crucial puzzle piece, eagerly claimed the final bow.
With Franklin's clues finally in hand, the true architecture of the molecule was unmasked, the double helix. It's essentially an elegant 3D spiral staircase, or you can think of it like a ladder that's been twisted upon itself. The sugar and phosphates link together in a continuous chain to form the twin backbones, the sturdy sides of that twisted ladder. This structure is beautifully robust, allowing it to coil up super tightly while keeping the precious information inside completely protected.
So the absolutely crucial point here is what actually holds the two sides of that twisted ladder together. Those are the nitrogenous bases, which act as the rungs. They lock together via relatively weak hydrogen bonds, but they play a highly strict matching game. Think of it like a biological lock-in key. Adenine, or A, only ever bonds with thymine, T, and guanine, G, only ever bonds with cytosine, C. This strict matching rule is the literal bedrock of all life on Earth, because that specific, sequential lock and key code is exactly what creates your unique genetic blueprint.
But this architecture has one more brilliant trick up its sleeve. It's anti-parallel. Chemically, the two backbones run in opposite directions. Think of it like reading a book. If the top strand reads perfectly from left to right, from what scientists call the 5' end to the 3' end, the complementary bottom strand is entirely upside down and backward, reading right to left. Because of those strict lock-and-key pairing rules we just talked about, I can challenge you right now. If I give you the top strand AGG-TCGG, you automatically know the complementary bottom strand just has to be TCC-AGGC. They complement each other flawlessly.
Now, understanding the structure is really only half the mystery. The real magic is how this massive biological code manages to copy itself billions of times over your lifetime without just completely falling apart. Your cells are constantly dividing, creating new skin, repairing muscles, generating blood. And every single time a cell divides, it needs a pristine, perfectly copied, 10,000-book exact replica of your DNA.
To pull this off, your body employs a highly coordinated mechanical replication factory. First, an enzyme called helicase comes in. Think of helicase like a rebellious teenager. It literally unzips the genes, slicing right through those hydrogen bonds to pry open the double helix. Once it's unzipped, RNA primase acts as the advance team, laying down a short starting line. Then comes the meticulous architect, DNA polymerase, which builds the new complementary strand by adding matching nucleotides. Finally, the master welder, DNA ligase, comes in at the very end to glue everything securely together into a seamless molecule.
But remember that anti-parallel, upside-down structure we just talked about? Well, that causes a major traffic jam during this process. We have what we can playfully call the good guy leading strand and the jerk lagging strand. Because our meticulous architect DNA polymerase stubbornly only builds in one specific direction, copying the top leading strand is a total breeze. It continuously follows the helicase unzipping process, building smoothly left to right. But the bottom lagging strand, it runs the opposite way. It's literally like trying to read a book upside down and backwards while someone is rapidly flipping the pages on you.
Because the polymerase has to frantically work backwards against the unzipping direction on that lagging strand, it has to build in these short, disjointed chunks. It is an absolute biological hack. These chunky sections are called Okazaki fragments. They were named after a brilliant married couple of scientists who added their crucial chapter to our century-long detective story by cracking this chaotic code back in 1960. So the primase constantly lays down new primers, the polymerase builds a tiny backward segment, jumps forward to do it again. And finally, the ligase welder glues it all together. It is a start-and-stop mess for sure, but it works brilliantly.
Now, we absolutely must acknowledge a crucial supporting character in this whole process, RNA. It's DNA's chemical cousin, but it acts as the unsung hero, the vital scout that has to go in first to prep the site so replication can even happen. It differs from DNA in 3 key ways. It's single-stranded, meaning it lacks that double helix stability. It uses ribose sugar, bringing an extra oxygen atom to the party, and it uses a slightly different chemical base called uracil instead of thymine. Without RNA venturing into the unzipped molecule first to lay down that primer, DNA couldn't replicate at all.
What really illustrates the sheer refinement of this biological machinery, though, is its precision. When your body is dealing with 6 billion letters of code, you'd expect a lot of typos, right? But DNA polymerase also acts as a rigorous proofreader. If it accidentally pairs an A with a G, it literally cuts out the mistake and fixes it on the fly. Thanks to this, an error only occurs roughly once in every 10 billion nucleotides. Without this hypervigilant proofreading, our genetic code would degrade rapidly, causing massive mutations that would make human survival completely impossible. It is literally keeping you intact.
So after nearly 100 years of scientific sleuthing, from washing pus off dirty bandages, to firing dangerous X-ray radiation, to deciphering the chaotic start and stop rhythm of Okazaki fragments, we finally understand the mechanics of the most famous molecule of all time. But I want to leave you with a rather provocative thought to ponder today. We know the exact structure. We completely understand the replication machinery. But when you look in the mirror, how does this microscopic, 6-foot-long, incredibly precise chemical puzzle of 6 billion letters actually construct your personality, your quirks,